cell proteins, or HCPs, are an inevitable byproduct of biologics manufacturing, and they can have an impact on the product quality and efficacy, as well as patient safety. Processes designed to remove HCPs from the bioreactor harvest have to achieve acceptable limits, but also be scalable. Our customer had 1 month to reduce the presence of HCPs in the bioreactor harvest prior to production runs that was going to supply drug substance for a scheduled phase 1 clinical trial. The level of HCPs in the downstream product was too high, 700 ppm, and this negatively impacted the plant clarification process and subsequent downstream steps. Our goal for this customer was to reduce the level of HCPs to less than 300 ppm and ensure clarification of the entire 2,000-liter harvest. We can use a number of options to reduce HCPs, such as mixed mode or hydrophobic interactions columns. For this project, these approaches were too complex and would not allow us to deliver an optimized solution within the compressed time frame. These options will also not solve the problem of clarification filter filtrability of the 2,000 liter. A reduction in bioreactor size to 1,000 liter, which will allow for clarification, was not an option due to the amount of drug substance needed for the trial. Given the shortcomings of the option I have just described, the approach we selected to drastically reduce HCPs and increase filtrability was caprylic acid precipitation, followed by filtration using Clarisol filters. We use design of experiments, or DOE, to identify the optimal conditions for precipitation with a focus on 2 key parameters: the concentration of caprylic acid and temperature. DOE enabled us to reduce the number of experiments required to evaluate these parameters, which was crucial as we had a limited number of cell bioreactors available for the study. Most importantly, yes, we did meet the 1 month time frame. The combination of caprylic acid precipitation and Clarisol filters enable a reduction of HCP to the desired goal of less than 300 ppm and clarification of the entire 2,000 liter harvest. And use of the Clarisol filters also had a positive impact on the downstream steps. It allowed us to reduce the number of filters needed, reduce the volume of water needed to wash the filter, and reduce the dilution of the material after the wash. With a reduced dilution, the capture state following the filtration required less time. What is also quite interesting about this project is that we strategically combine an innovative product, our Clarisol filters, with a conventional approach to precipitation.